Breeding transgenic animals allows for the process of reverse genetics, the discovery of the function of a gene by analysis of the phenotypic or observable effects of the specific engineered gene sequence. Let's see how to make transgenic knockout animals, or animals that do not express a particular gene. Although transgenic animals can be any animal for which we know the parts of its genome, the most commonly used is the mouse. The mouse gene that we seek to stop expressing is isolated within the mouse genome. We then mark it with the neomycin antibody gene by creating a replacement vector. This vector contains the beginning exon and the end exon for the gene, and the gene for the neomycin antibody in the middle. This neomycin antibody gene allows the cells to express neomycin antibodies. Thus, cells expressing the marked gene survive, even in the presence of neomycin, which usually destroys growing mouse cells. The vectors are injected via electroporation in mouse stem cells from the fertilized zygotes. Neomycin is injected in the growth environment of the stem cells. Neomycin kills all the stem cells, but the ones that have incorporated the gene marked with the neomycin antibody. The surviving cells are re-injected in their blastocysts of origin. The blastocyst is then allowed to grow in a host mother. The resulting offspring is a chimera, containing some cells that are knocked out and others that are not modified. This organism's offspring will be fully transgenic, with the gene of interest knocked out. This transgenic mouse may not survive the gene change, showing the gene is too critical to survival to be removed. Let's imagine that the genome is a piece of music. Knocking out the gene for measure lines may result in the organism replacing them with double bar lines. This results in an organism that is incapable of expressing critical parts of the music piece, and thus the song cannot be played. The mouse may survive without any phenotypic effects, showing the mouse may have developed a compensatory mechanism in the gene removal. In this mouse, the knocking out the measure lines did not affect the music piece because it relies on the beat fraction to survive. In some cases, the mouse shows phenotypic effects that can be attributed to the gene knockout. For instance, knocking out the ability to produce an open D affects the overall melody, or phenotype, but the music can still be played. Learning what the phenotypic effects of a gene removal are may help us understand a biological mechanism, associate the symptoms of an illness with the modifications of a specific gene, and project possible causes for symptoms.